to Coffee and Community, Cutaneous Lymphoma Conversations, brought to you by the Cutaneous Lymphoma Foundation. I'm today's host, Holly Preeb, and I'm glad to have you with us. Joining me today, we have Dr. Stephen Horwitz, medical oncologist on the lymphoma service at Memorial Sloan Kettering Cancer Center, where he leads the T-cell lymphoma program. In addition to his many professional accomplishments, Dr. Horwitz is also one of our dedicated board members, as well as a passionate volunteer for the foundation, lending his expertise across many of our programs and services. So grab your favorite cup of coffee, settle in as Dr. Horwitz and I dive into the recent Lymphoma Research Foundation T-cell workshop that the Cutaneous Lymphoma Foundation proudly partnered to bring the cutaneous lymphoma sessions to. Welcome, Dr. Horwitz. Thank you for joining us this evening.
Thanks, Holly. It's a pleasure to be with you. Thank you.
Well, you are a man of many hats. Tonight, you're joining us as the T-cell workshop co-chair. So I thought I would start by asking you, what exactly is a T-cell workshop?
Yeah, so it's a good question. I'm sure they'll all be good questions. So there's a lot of medical meetings that we have, and I think they take on various forms or various agendas. I'd say the two most common types of meeting we have are educational meetings where people get updates on a topic, sharing information we know to the audience so they can take it back and apply it to their practice, or if it's patients, so they can learn about their disease. And then there are scientific meetings where people present new research to disseminate their research and new ideas, again, to the wider academic community. And again, that's usually the speakers presenting. With the workshop, the goal was really to bring not all, but many people in the world who work on trying to understand or find better therapies for T cell lymphomas together and really have speakers introduce topics, but then have a lot of back and forth with the audience with the goal of trying to bring people together, a bit of level setting to understand where we are in the field. I can explain more about that. And then really try to think of agendas and issues and resources and collaborations that the group together could work together on to move the field forward. So it's really workshop, meaning it's work. It's information, but the idea is that there would be an output of things we want to accomplish and how we would go about accomplishing that. So really very agenda driven in that sense, if that makes sense.
No, absolutely. I think that provides really good clarity on the T-cell workshop. So why add the cutaneous lymphoma specific sessions to the overarching T-cell workshop?
So the theme of the workshop was biology to therapy, and what that represented, which is probably, as your audience knows, therapies or effective therapies or curative therapies in T-cell lymphoma are less than they are in some other cancers, and certainly less than they are in like B-cell lymphomas, which is a very analogous. The understanding of biology, how these lymphomas develop, what happens when they develop, what are the things going on in the cells, has really accelerated a lot. Really, in the last 10 years, we've learned a ton about the biology of these diseases. It's always slower to translate that to therapy, but the idea was, how do we take those biologic advances and use that to set the stage to develop, identify better therapies, better combinations? Not everyone totally agrees, and there was some conversation at the workshop, but when we look at the biology, or when I look at the biology, there's a lot of commonalities because they're cancers of T cells, whether they present in lymph nodes or blood or bone marrow or the skin. And therefore, the understanding of biology really can cross the spectrum in a bunch of different T cell lymphomas, including cutaneous T cell lymphomas. Now, because the cutaneous T cell lymphomas are different in how they affect people and how we approach therapy, and the fact that a lot of them are on the skin, which is on the outside, which lends itself to therapies like light and radiation or external therapies, how we apply those therapies is different. So the first part of the meeting was really pathology and biology, which largely crossed systemic and cutaneous T cell lymphomas. And then when we move on to therapy, that's where we get into some specifics of how you would apply those therapies to cutaneous T-cell lymphomas, how you might study those therapies. I oversimplified a little. There are some unique biology or different aspects or different studies. So we put that as an additional or a separate session to really focus on that. And then I think the practical reason was we had speakers from, around the world coming, and the audience isn't exactly the same. So there are dermatologists who participate who we didn't know whether they would come for the whole meeting or only that. So by cohorting that group, we also could make it easier for some people traveling who have a specific skin focus to participate. But kind of for all those reasons, that's just kind of how the design of the workshop unfolded.
That's really exciting, and it explains the difference between the cutaneous lymphoma across the general T-cell lymphomas as well. Appreciate the extra explanation. So, is this a one and done? Is this something that, needs to happen, but only needed to happen once? And
Yeah, I hope not. The Lymphoma Research Foundation has had a series of workshops that some have gone on for many years, I think some a few decades, where people have gotten together to focus on specific subtypes of disease. And those workshops have different forms. Some are really agenda driven like this, some are more updates. But I think our hope was that we will set the agenda, take away action items. We already have an emerging list of things we still want to accomplish and work on for future workshops, but hopefully take all that together and then move forward and make this a recurring event. Whether it's yearly or every other year, the fields only change so fast. But I think we'll see how that evolves. I thought the event was great. There was tons of enthusiasm. We had over 100 people from 12 countries, and we didn't even have everyone come. So I think there's really an opportunity to develop momentum, and hopefully that can be ongoing. There has to be interest, there has to be some output and success, there has to be funding. So, we can't just declare we'll have this and automatically make it happen. But it seems like there's a lot of enthusiasm. And particularly when you think about a rare disease, some of the barriers to moving the field forward are collaboration and working together, because in rare diseases, getting enough patients or patient samples or resources together can be a challenge. So some of the things we're working on now as a follow-up to the meeting is how do we bring some of those collective resources together? And I think if we're able to capitalize on that and move things forward, then I think we will be successful in having this a recurrent thing and truly try to accelerate progress kind of across the board in these diseases.
that's wonderful. So, are you able to share any of the discussions or topics that came up, or things you learned or outcomes with our audience?
Yeah. Yeah, I mean, some of it's a little technical, but what we had, the basic sessions were initially was a pathology session, so diagnosis and what we understanding about the diseases. And we had, again, speakers from around the world who were experts in different areas of T-cell lymphoma present what's sort of understood about the pathology. Then the next group of sessions were translational science. So we had speakers on techniques to try to understand what's going on in individual cancer cells or in cancer lesions, both the cells and the cells around the cells, and techniques and technologies to sort of interrogate that and set the stage for new therapies. And then we had speakers on some of the different themes of new therapies. And then we had speakers on clinical data. So, what are the results of early clinical trials of different types of targeted therapies, and then cutaneous lymphoma as a wrap up, which kind of brought together a lot of cutaneous lymphoma. So, without going too long, but probably going too long, we asked each speaker to sort of, structure their talk as they thought. But the idea was to talk about not a complete review and not necessarily all their new research, but big questions like what do we know? What do we not know about whatever their topic is? What do we need to know to make the next advances? And then how we might go about answering those questions to move forward. So, for example, like one thing in pathology, what we talked about is that the pathology classifications have evolved historically, initially from what cells look like under the microscope in T-cell lymphomas, also where they are in the body. Are they in the skin? Are they in the nasopharynx? Are they in the liver, lymph nodes? And that's how the diagnoses emerged. We now know a lot about the biology. So some of those groups of diseases are clumped together because we see, convergent phenotypes. So different diseases or maybe similar and lumped together because of how they appear in the body. But when we really understand what's going on at the cellular level or things like that, there's already, we know there's different patterns. And within each group of lymphoma, we know there's subsets. So for this audience, Sézary syndrome is not just mycosis fungoides in the blood. There's biologic differences, they overlap. And large cell transformation or tumor stage MF is biologically different than patches and plaques. We may call it the same disease, but as we move beyond radiation and chemotherapy, understanding new therapies need to probably require understanding what's going on in those cells. So, one thing we talked about was how do we work together internationally on pathology, sharing samples. And one idea that came up was that imaging quality is so good on slides now, and our center's been scanning slides for years, and other centers do that. That could we start off by sharing images to have pathologists from around the world not look at 100 cases, but look at 500 cases? And can we use that imaging to sort of understand differences within these subtypes? And then can those differences help us understand the words I would say would be vulnerability? So, are there things about those cells that make them vulnerable to different kinds of therapy? And how could we then understand that, who those patients are that would most benefit from those therapies, who the patients who wouldn't benefit from those therapies are, and then apply the optimal therapy for each patient? That's a multi step process, but that's, for instance, like one example of something that we could work together on that. If I keep going, other topics were unique. Other vulnerabilities in T cell lymphomas based on some things like the nutrients that those cells need, understanding when immune therapies work and don't work. So I don't know if your audience may know that we use immune therapies or what we call checkpoint inhibitors for lung cancer, melanoma, many kinds of cancer, and they're very effective. Some colon cancers or rectal cancers, so based on different things in the cells that make them sensitive to stimulation of that person's immune system. So in T cell lymphomas, it's been a little bit complicated because most of those checkpoint inhibitors try to stimulate and activate good T cells to fight cancer cells. And with T cell lymphomas, you have cancerous T cells as well as good T cells. And how do you understand that? Because when those therapies have been studied in patients, we've seen both some people respond and other people can have their cancer cells stimulated and have something what we call hyperprogression. So it almost drives acceleration of the malignant or the cancerous T cells. So there was talks now, how might we separate that out? What are some of the features in the cells that might start to tell us that that cell would be a good target for stimulating good T cells to fight it? Or are there things in that cell that might tell us that that cell would be susceptible to being itself stimulated or growing or driving? So how can we start looking at that? Because that both benefit and high risk have really hampered us moving forward with some cell therapies or some immune therapies in T cell lymphoma. I could just keep going. I mean, it was a full 2 day session. So, are there therapies that work on specific pathways? The JAK-STAT pathway is another what we call a signaling pathways. Basically, with a lot of T cell lymphomas, normal T cells have something on their surface called a T cell receptor. And that T cell receptor gets activated by recognizing something that shouldn't be in the body. That's how the immune system works. So if you have a flu or something like that, the T cell receptor may recognize an infection or a virus. And if it meets that virus, it tells that T cell to divide, become activated so it can function as a normal immune cell, reproduce itself. That's how the body makes a bigger immune response to, for instance, fight off infections. So we think a lot of T cell lymphomas have kind of signaling through the T cell receptor, where instead of the T cell responding normally when it encounters an infection and then sitting there quietly when it doesn't, it's turned on all the time through various reasons. And some of those processes inside the cell that turn it on, some of those are mutations, some of them goes through pathways, something called the JAK-STAT pathway, which is fairly common in a lot of different T cell lymphomas. So it's like a signaling pathway that should only be on when it's supposed to be on, but it might be abnormally on all the time in T cells, making them grow and divide or not die and persist. So there was talk about a lot of that because that's one of the pathways that we think we can target. How do we understand which cancers are most susceptible to that, which ones are not? What are the better inhibitors or ways of targeting that pathway? How you might combine that, like when cells don't die by targeting that pathway, what would be the other mechanisms you can target? So, kind of things like that. It's a little technical, but trying to understand that, what we understand about the biology, understand what we've seen when we've tried these therapies in people, and then trying to match that up like, okay, so this is a group or these are diseases or these are features in the cell that make this a good therapy. When that works, how do we build upon it to optimize it to make that therapy as effective as possible? So it was high level research, but it was great. And then there's a lot of back and forth, and people from the audience who also study these things ask questions. What about this? We've seen this. Okay, how might we look at answering some of those things?
It sounds like great discussions. It sounds like lots of crucial work being done to try to target and understand these subsets of cancers and what's happening going forward, or how do you develop the plan for going forward. So with all of this that you started, you mentioned that there's a need for additional. What would be a next step? What would you hope to do at the next workshop, to learn, to take from your outcomes here, to move forward?
Yeah. I think there's kind of probably two big buckets for that. There's one is what did we start here that we can advance? So, you know, these are complicated diseases. If it was easy, we would have better therapies more often, things like that. And they're rare diseases, so they tend to be understudied. So, how are we mobilizing groups together? What have formed to sort of resources where people can work together to answer some of these questions? So, updates on that. What are the next steps? What are barriers we've encountered? Because none of this will be totally simple and straightforward. And then, how do we problem solve those barriers? And then, I think as we learn more about biology and we test new therapies, secondary and quaternary questions will arise. Okay, now we've done this, we see this and this. So, why is this not working here when we think it should? And how do we then set it up to answer those questions? How can we work together to do clinical trials in patients of rare diseases across more centers when pharmaceutical input and other funding is less than it was? So, in diseases that are common, there's often a significant pharmaceutical input in terms of supporting funding, and that can drive an agenda. We haven't really had that in T cell lymphoma because it's quite rare. So we have a little bit of that, but it's more often been on the academic centers to work together to drive the agenda. So to make sure we're staying on task and we're moving things forward and kind of optimally working together. And then I think there were other questions that we just, the meeting was 2 full days. We just didn't have time to cover everything. So one thing that came up was interacting with the FDA. So in these rare diseases, sometimes the standard approval processes don't work that well because you can't do huge randomized studies all the time with hundreds or thousands of patients. You're often trying to understand what therapies work and don't with smaller numbers. And I know Cutaneous Lymphoma Foundation has been involved in this with and other rare disease organizations, the Lymphoma Research Foundation. So we talked about having a session inviting the FDA and talking about other ways or additional ways to study outcomes in new therapies of rare diseases using real world evidence. There's things called synthetic control groups. So you don't have to expose as many patients to therapy that we already think is not that good in a randomized study, and ways to sort of optimize or streamline clinical trials. We kind of decided that this agenda for this meeting was already pretty full, and that was almost a second step. Like this was kind of getting the community just more on board working together, and then bringing clinical data to the agency is probably a later step. But that was something we would want to have. Several subtypes of diseases, some of the virally driven lymphomas, some caused by HTLV1. That's probably a whole nother session of biology. We just couldn't do it all in this session. So my vision would be the next meeting probably is not too long, maybe a year, maybe a year and a half, something in that time frame to cover some of the topics that we didn't cover, and then do a short interim update for some of the agenda items that we laid out for this meeting.
That's wonderful. I mean, I hear so much hope in the things that you're talking about, in addition to the knowledge and expertise that you and your colleagues are gaining from each other, the potential for new therapies as you think kind of outside the box in how can we work with the regulatory bodies to ensure new therapies are coming to market for patients, which is amazing. And I know will bring a lot of hope to the people that we serve at the Cutaneous Lymphoma Foundation. So, to that, I think I remember one of the outcomes was a kind of a roadmap that you had developed. Is that inclusive of all the things we've been talking about, or is there more to that roadmap that we should include here?
Yeah, I think there's both more and less. So I think there was, we just had the meeting, just finished up. There was a lot of sort of questions and next steps. So what we're doing now is kind of debriefing, getting some more like formal feedback from the audience, getting together and kind of listing sort of all the many, many ideas that came up, the many ways of working together. And then I think what we're going to do is try to streamline that into, okay, we think these are the immediate next steps. We think these are things that are accomplishable in a reasonable period of time. I think some of the ideas probably are still waiting on further understanding to how to apply, or different therapies that maybe don't exist yet for how to apply them. So I think we'll kind of collect all that, and I think over the next few weeks to month or so, write something out that kind of more streamlines. Okay, this is what the group thinks are the next steps. This is how we might accomplish that. Then send that back out to the group, kind of get feedback, and then sort of get buy-in on strategies and goals. So yes, but it'll take a couple steps to sort of formulate that in a more clear-cut and agreed upon path.
Yeah, sure, for certain. So, knowing that you are speaking to the patient population with this podcast, from your perspective as an outcome from this, what's important for the patients to know about the work that you and your colleagues are doing on their behalf, and, where you're trying to get to?
Yeah, I think the hope message I think would be important because a lot of people are really interested in working on it and bringing energy and intelligence and ideas to do it. So I think that's coming. I think progress for cancers like this is never fast enough. But I think in the past we've sort of maxed out on the what I might say are sort of the more agnostic therapies. So chemotherapy, radiation can kind of kill dividing cells somewhat indiscriminately. So we already know where those work and where they don't. There's probably limits on how far you can advance that. And then, really, all the newer therapies and the advances are really going to come from understanding the biology of what's going on in the cancer cells. And then, how do we address what's wrong in those cells or what's wrong in the immune system that it allows the cancer cells to grow and flourish where the body should be taking care of them. So, I think that understanding is already way advanced than it was 10 years or 5 years ago. So, I think that's the hope, and then we just have to kind of bring that together, probably learn a little more, and then continue to like take our best ideas and see how they work in people, make sure they're safe, understand where they work, understand where they don't work. That's a little bit like hand to hand combat in sort of slow. Like you can't just have a big idea and make a therapy work great. You have to try it out and then understand where it works and where it doesn't. One of the big topics in the meeting was heterogeneity. So T-cell lymphomas are heterogeneous, so there's a lot of differences in individual differences. So understanding that, but then just sort of continually take what we learn, move it forward, try that out, what we learn from that, filter it back into what we just learned. Is there a better combination? Is there a better way to match patients to therapy? And then just continue to move forward. So I think that forward progress is there. Again, not fast enough, but there's enough collective work and enough good ideas and good understanding that I think we'll just keep moving forward from here as fast as we can.
Oh, absolutely. And I know that the members of our community so appreciate the work that's being done on their behalf. So, to that, people who have cutaneous lymphoma and people who have loved ones who have cutaneous lymphoma are always interested in how do they play a role, what can they do, how do they make a difference in their long-term plan, and how do they support the clinical community? What would you say to somebody who reached out to say, I want to help? Some might have funds, some might not have funds, but how do they help support advancing research and these types of efforts of collaboration amongst your peers?
Yeah, I think there's a lot they can do. I think just, inspiration and support and being part of the community, attending meetings, asking good questions, educating themselves. I think that's great. I think advocating for this group of diseases as well as rare diseases in general in terms of keeping momentum, getting attention from pharmaceutical or biotech companies. We're in a period now where I think some government funding for cancer research is unclear where that's going. I think we've heard a lot of stuff, but it's sort of unclear. So I think conveying the importance that, particularly for these rare diseases, it's going to be academic centers, government funding, national cancer, things like that really do a lot of the initial work because these are not huge market blockbuster therapies for these diseases. So I think support and advocacy that these are things that are important for our society to support, important in people's lives. I mean, certainly financial support to Cutaneous Lymphoma Foundation, Lymphoma Research Foundation. This meeting was funded by donors. It was not primarily funded by pharmaceutical companies. So I think when people have means, those things can help. And then when it's appropriate, at least considering clinical trials, it's never something that I would say you should do to help the field. But there are times in your disease course where it might be the right thing for you. And as an important side effect, it also helps the field. So I think understanding where those things exist, being open minded to it, always, of course, making the best decisions for you and your family, but sort of understanding that that's one of the ways that research moves forward is with patient participation. If you're asked to donate an extra sample of blood or part of your skin biopsy to research, again, if that's safe and comfortable for you, those are samples that many centers like ours collect and use as part of these research efforts and understanding. So, I think just being aware and seeing where your energy or your interests aligned with what the research community is trying to do.
Well, that's excellent. Those are all really valuable ways that people can get involved, and it spans, all kinds of people, their different capacities. So I appreciate you sharing those. What did I miss? Did I miss anything, talking about the T-cell workshop, things that would be important for the community to know about the effort?
Yeah, I think you really got the highlights, and I think probably the main message, because so much of it is sort of technical in the biology and a little bit into the weeds of how are we going to tackle these things. But to me, the headline was more than 100 people from around the world getting together, devoting their time, really focusing on understanding these diseases and really the spirit of like wanting to work together to accelerate progress. So to me, that's the main thing that I took away from it too. Each of these individual problems, we have to work on and figure out, and those will, be solved at different rates based on, you know, what we, understand and how fast you can move forward. And, some problems just end up being harder to solve than others. But I think the spirit of collaboration and momentum, to me, that was really the highlight.
I think that's great. I really want to thank you, Dr. Horwitz, for your time and sharing of your knowledge and your expertise once again with our community. It brings us to the end of Coffee and Community Cutaneous Lymphoma Conversations. We hope that you, our audience, found this discussion insightful and inspiring, the same as I did. Before we go, one last thank you for Dr. Horwitz for his commitment, as well as a thank you to our sponsors and donors. Without their support, programs like these would not be possible. Remember, you can find more episodes, resources, and a full list of our sponsors on our website at www.clfoundation.org. If you enjoyed today's episode, please consider subscribing, rating, and leaving a review. Your feedback helps us reach more listeners and continue bringing valuable conversations to and from the Cutaneous Lymphoma Foundation community. Thank you for joining us today. Until next time, stay informed, stay connected, and take care.